a founding member, principal scientist in Expansion Technologies Incorporated, Cambridge, USA. He is now head of R&D Expansion Technologies Incorporated, Cambridge, USA.
Hello.
That's it, the introduction.
Yeah. Thank you, Johnny. Over to Dr. Mahender Dewal. Sir, I think you are there. You can unmute and.
Hi.
Yeah. Hi.
Good morning. Good morning, everyone. Let me share my presentation first, and then I'll start talking. Okay. Can everyone see the screen?
Yes, sir. We are able to see.
Can you hear me, everyone?
Yes. Yes. So we actually,
come on, please.
We actually muted all speakers, so we are able to see. Yeah.
Yes or no?
We actually muted everyone, so everyone able to see and hear you.
Okay. What's going on here? Let's see.
Yes.
Hello. All right. I think everyone can hear me and see my screen. Okay. Give me 1 second. Let me share it again. Okay. Good morning, everyone. Thank you so much, Johnny, for your nice introduction. Thank you. First of all, I would like to thank Youth for Inclusive and Sustainable Society members for inviting me today to give this talk. And I would like to thank all the members, including Mr. Kiran Kumar and his team, for all the hard work and efforts putting this event together. Thank you. Thank you so much. So in today's talk, I try to keep it as general as possible, but after the talk, I'm happy to discuss as much as scientific depth as possible to make sure that everyone gets the point. So in my talk today, first, I'm going to talk about, I think my slides are not moving for some reason. Give me 1 second. All right, okay. So in the first half of the talk, I'm going to discuss about the evolution of coronavirus and its infection mechanism. Then I'm going to briefly discuss about the immune system, the components of the immune system, how it fights with virus infections. In the second half, I'm going to discuss about the various different methods of therapeutic and vaccine developments and current progress in their developments. COVID-19, 5-letter and 2-number word, has been shaking the world from past 6 months, crazily. What is this COVID-19? COVID-19 is an infectious disease caused by the most recently discovered novel coronavirus, which starts with most common symptoms: fever, dry cough, and tiredness. Recently, Most of the patients, approximately 70% of the patients reported two additional symptoms, which is losing the smell and taste. Therefore, most of the countries now included these two characteristics as identification marks for the COVID-19 infections. In most severe cases, COVID-19 can cause pneumonia, severe acute respiratory syndrome, SARS. That's why it's also called SARS coronavirus, leading to organ failure, which includes kidney, lungs, and other organs, ultimately leading to the death. When I said novel coronavirus, this COVID-19 is caused by novel coronavirus. What is novel about this? Is there any other coronavirus already existing? Let's take a look into the history a little bit. Human coronaviruses were first described in 1960s. Here I have listed 4 different coronaviruses that were described in 1960s, which cause mild symptoms with the common cold. And these are also not considered as highly pathogenic viruses. It only caused mild symptoms in immunocompromised patients. So these coronaviruses are originated from either bats or rodents and transmitted to the humans via an intermediate host, such as camels or cows. However, in November 2002, SARS coronavirus 1 broke out Fushan province in China, which spread to 5 continents, approximately 30 countries. WHO announced it as an epidemic, not a pandemic, because it is limited to certain region. And the source of this particular coronavirus is also bats, but the intermediate host is civet cats or raccoon dogs, which causes SARS pneumonia from mild to severe, mild with severe symptoms. Approximately 8,000 people from these countries were infected by this virus, having 800 of them declared dead. Also, in 2012, another coronavirus, Middle East Respiratory Syndrome coronavirus, MERS coronavirus, broke out in Saudi Arabia. This was also an epidemic which spread into about 27 countries. And the origin for this particular virus is also bat, but the intermediate host is dromedary, which is also called Somali camel. MERS coronavirus causes MERS or acute respiratory distress syndrome, which also symptoms include mild to severe symptoms with a lethality rate of 35%. Approximately 2,000 people from these countries infected by this particular virus, and 700 of them are declared dead. As most of you know that in December 2019, from Wuhan province, China, new novel coronavirus broke out, and within few weeks, it became a global pandemic by March 2020, announced by World Health Organization. And this novel coronavirus is highly homologous to SARS coronavirus 1. That's why it is also called SARS coronavirus 2. The origin for this particular virus is also bats, and the intermediate host is different from compared to the other coronaviruses, which is pangolin. The disease is caused by this coronavirus is COVID-19. As of today, approximately 9.64 million people are infected by this particular virus all over the world, having almost 500,000 people are dead so far. So this, if you look at these 7 generations of coronaviruses, last 3 coronaviruses are kind of more infectious and causing some severe symptoms compared to the first 4 generation coronaviruses. Next slide, I'm going to discuss about similarities and differences between these 3 coronaviruses, SARS-1, SARS-2, and MERS. One of the most important similarities is all these 3 SARS coronaviruses are originated from bats. That's why the genomic information is 90 to 95% is similar with bat coronaviruses. But the SARS coronavirus 2 and SARS coronavirus 1 genomic information matches approximately 80%. And the SARS coronavirus 2 is 50% genomic information is matches with MERS coronavirus. The SARS-1, SARS-2 belongs to pretty much close relatives like brothers, but MERS is kind of a cousin to the SARS coronavirus 2. Another important difference here is the cell entry receptor for SARS-1 and 2 is same, but MERS is different, which is ACE2 for the SARS-1 and 2, which is called angiotensin converting enzyme 2. But for MERS, DPP4, dipeptidyl peptidase 4. So in comparison with SARS-1, 2, and MERS, SARS-2 became so infectious, very rapidly spreading. Let's take a look into the reasons why this is so rapidly spread. So the primary cause of the spread for this SARS-2 coronavirus is person to person. By coughing, when an infected person coughs or sneezes or speaks loudly, the virus particles come out of the mouth and infect the other persons. And it also has a secondary spread reasons. One is airborne transmission. The virus filled droplet particles that came out of the infected person can survive in the air approximately 3 hours, which can infect other persons as well. And one more important thing is contaminated objects or surfaces also can transmit this virus infection to the uninfected people. If you look at the table that I listed here, the survival rate of the virus at 4 degrees centigrade on various different surfaces is approximately 2 weeks, which is in the fridge. And at 37 degrees centigrade, approximately at the body temperature, not inside the body, but outside the body, it can, it can be detected approximately 1 to 2 days. But in hot water, 70 degrees centigrade, within 5 minutes, this virus will be cleared out. And another interesting feature is if you look at the regular paper and tissue paper, the survival of this virus is approximately 3 hours, 30 minutes to 3 hours. But if you look at the currency note and cardboard, which are also made of a paper, the survival is quite different from 2 to 4 days. This is quite unique about this particular virus, and why this is behaving like this, the data is not clearly known, but these are also causing rapid spread of this virus to uninfected people. And another important feature of infection speed is long incubation period. The incubation period for this particular virus is 2 weeks. The infected person may not even show the symptoms up to 2 weeks, and then he may continue to spread the virus to the other people without knowing himself. And recently, one paper published about the mutations. This particular mutation, an aspartic acid to a glycine at 614 position of one of the proteins, which is spike protein in the coronavirus. This mutation, when the virus gets this mutation, also spreading rapidly its infection. And this is this work is done in the laboratory cultured cells, but it's when the correlation studies are compared with the genomic data available in the data banks, which matches from January to till June, the virus that is detected with this glycine mutation is much more 70% higher than the aspartic acid, the original virus. So what is this coronavirus? Let's look into the structural features of this coronavirus. Once, Peter Medawar is a Nobel laureate and immunologist, defined in general virus as simply a piece of bad news wrapped up in proteins. Exactly. Like living cells, viruses contain genome. It could be a DNA or it could be an RNA. In the case of coronavirus, a large single-stranded positive RNA is the genome of these coronaviruses, which is securely coated by a nucleocapsid protein. If you look at here, another unique feature is this nucleocapsid protein secures the virus RNA, and it has a nice structural features. You can see this spiral structure, which is not common in other RNA viruses. This is unique for the coronaviruses. And other important feature is these spike proteins, which is not common with other RNA viruses. And because of these spikes, they look like a crown. That's why it is called coronavirus, which the structure also has all these structural proteins: spike, envelope, membrane, and nucleocapsid. Unlike the living cells, the viruses does not divide 1 to 2, or like human cells, 1 to 2 or 2 to 4, 2 to 6. They depends on the host cellular mechanisms to replicate itself. And this coronavirus belongs to a family of zoonotic viruses. That means the virus originates from birds or animals, but it can transmit from birds or animals to animals, and animals to humans as well. Now, let's take a look into this spiral genome a little bit much more closer so that we can understand the virus better. As I said, it has a large positive sense RNA, which approximately 29 to 30,000 nucleotides, encoding 29 proteins. Here is the genomic map displayed here, which can be divided into 2 parts to understand this better. In the first part, it encrypts the information of 16 non-structural proteins. That means they are very active in the function of the virus. and the rest of the part has 4 structural proteins as well as the 9 accessory proteins. The functions of these non-structural proteins, among the 16 non-structural proteins, only 3 of them are very well understood as of. These 2 proteases, which plays an active role in virus maturation by cleaving the long polypeptide chain of the viruses. The other enzyme, which is RNA-dependent RNA polymerase, one of the unique enzymes that this virus has, which error-proofly, without any errors, transcribes the virus positive sense RNA into a negative sense RNA, which cellular enzymes or makes the more and more virus RNA. It basically acts like a template to make more RNA. And functions of accessory proteins, not clearly known. And the functions of structural proteins are very crucial, very important. As I mentioned in my previous slide, nucleocapsid protein secures the genomic information, the RNA. It bonds to RNA and it gives a proper structure to the RNA. And spike proteins plays very important role in cell enter into the cell. It binds to ACE2 receptor to enter into the human cell to infect the cell. Envelope protein works together with membrane protein. It interacts with it, and then membrane protein basically organizes the virus assembly, and together envelope protein and virus membrane protein together brings structural features to the coronavirus. Now let's take a look into the cell entry mechanism. As I mentioned in the previous slides, the spike protein attaches to the ACE2 receptor on the cell membrane. This is the cell membrane down here. Once it is attached to the ACE2 receptor, some of the cellular enzymes like proteases forms this nice endosomic structure, which is a process is called endocytosis. Once an endosome is formed, the spike protein takes some functional conformational changes and it fuses into the membrane. Then once it is fused inside the membrane, it basically releases the RNA into the cytoplasm of the human cells, like a revolving, like inside out, revolving, door revolving. And in this cartoon here, we have two, which clearly shows the interaction of spike protein in red and ACE2 receptor on the cell in blue color. The spike protein has two subunits. Here is a map here. First, this half is called S1 subunit and rest of the half is called S2 subunit. In the S1 subunit, this particular domain, which is called receptor binding domain, plays very important role in strongly binding to the ACE2 receptor, which initiates the cell entry. Once the virus enters into the cell and releases the RNA, the cellular enzymes start translating the viral positive sense RNA into all various different viral proteins, which includes the replicase. As soon as functional replicase is formed, it starts transcribing the positive sense RNA into a negative sense RNA, which becomes a template. And this template, the cellular enzymes uses this template to create more positive sense RNAs and the other RNAs of the other mRNAs of the other structural and accessory proteins of the virus, which will be then translated and transported by viral cellular enzymes, human host cell enzymes, and for example, the spike, structural proteins will be transported to endoplasmic reticulum and Golgi, where they get matured or refined, and then at the membrane of these Golgi, they started assembling. The RNA will come from the cytoplasm, and all the structural proteins are really here, and the other accessory proteins come from the other parts, and then the functional virus start taking an assembly. Once the functional virus assembles, it forms an endosome, and then the endosome comes to the cell membrane, and the virus will be released into the bloodstream. The process is called exocytosis. When virus infects a human cell or a new virus forms and it comes into the bloodstream, the very first immediate response by the human body is activating the immune system. This immune system is a surveillance system, which activates immediately when it sees a foreign object or a foreign infection. This immune system has two arms. First one is called innate immunity. This is the frontline defense. And innate immunity comes by birth. Each and every individual by birth will have an innate immunity. And this is non-specifically responds immediately to attack the virus infection or any other sort of infection within hours, which has many cells, different cell types, which I'm going to discuss in my next slide. And let's talk about the other arm of the immunity system, which is called adaptive immunity system. This is kind of a backup defense. It has a specific response. It activates, it doesn't come with by birth. It has to be developed as we grow, as the human grow. That's why infants, babies, and doesn't have a adaptive immune system, well developed adaptive immune system. And also these adaptive immunity system cells, their function, including innate immunity system functions, goes down as we grow older and older. That's why older people doesn't have a very active functional, even though they have the cells, but the functional immunity system, that's why they're also prone towards more infections. And this adaptive immunity system takes approximately a week days to activate, but once it is activated, it lasts for months to years to clear out the infections, that particular infections. Innate immunity system. So very first, the response by body is activating the immunity, innate immunity system. In here, all these cells, there are many different cells: monocytes, basophils, neutrophils, and eosinophils, so on, nature killer cells, macrophages, antigen-presenting cells. There are so many different cell types. They crosstalk each other, and then they release. There's signaling molecules which are called cytokines and chemokines, which basically helps the cells to crosstalk between themselves and between the infected versus non-infected cells and infections as well. So in this cell, I'm not going to talk about all the different cell types, but most important cell types are macrophages and nature killer cells and APCs. What macrophages does is basically they identify based on the chemo and cytokine signaling response. They basically engulf the viruses and clears them out from the system. Then when nature killer cells, they identify the infected cells and activates the suicidal mechanisms and then apoptotic pathways to get the cell itself killed. And as soon as the cell is dead, the macrophages eats it and clears them out from the system so not to make any poisonous molecules in the body. While macrophages and nature killer cells start doing their job, these antigen presenting cells will be activated. What they do is basically they find the antigens on the infected cells and infections itself, like viruses, takes them and run towards the adaptive immune system and gives those antigen representing molecules to the adaptive the cells present in the adaptive immunity. Adaptive immunity has basically two different cell types: B cells, B lymphocytes, T cells, and T lymphocytes. As soon as it identifies the antigens, T cells forms two different cell types: T helper cells and cytotoxic T cells. Cytotoxic T cells basically have the same function as the macrophages. They basically lyse the infected cells and clears them out from the system. They also generate memory cells, which plays very crucial and important role in disinfecting the viruses, killing the viruses in longer term. So these memory cells remembers the viral characteristic features of that particular infection, like that virus, and how to fight it. Then they completely clears up whenever the virus comes in, and then they clears up because they stay longer period of time. Beta cells and generates so many antibodies, including the neutralizing antibodies. The neutralizing antibodies basically they bind, in the case of coronavirus, they bind to the spike protein and inhibiting or prohibiting from the spike protein binding to the ACE2 inhibitor. And then the cytotoxic T cells come and clears out the viruses. This is how the body immune system functions fighting against the viruses. However, this coronavirus is somehow escaping the immune surveillance. How it is escaping, the detailed mechanistic details are not still known, because this is just 6 months old. It's been only 6 months. We already have solved so much information about this virus, but still a lot to know. There are some hypotheses available that I'm going to discuss in a minute. So when the virus enters into the body, it sends wrong signals to the immune surveillance system, which causes releasing so many cytokines molecules, which is called cytokine storm. And cytokines are two different types. One is pro-inflammatory and anti-inflammatory. These inflammation causes damage in the healthy tissue cells, which causes organ dysfunction, organ failure, leading to the death. That's how this virus escapes and causing severe symptoms in very people who has immunocompromised. So in some of the hypotheses, so researchers found that some of the accessory proteins may be helping the coronavirus to escape from the immune surveillance. And the other hypothesis is the spike protein of coronavirus is heavily sugar coated. That means heavily glycosylated. It has so many glycosylation sites. Probably that is protecting the spike protein detection from the immune cells. That's another hypothesis. The third hypothesis is conformation flexibility of the spike protein. This is the cryo-electron microscopy structure of spike protein. This is the subunits, and this is the receptor binding domain, which is in two different conformations. This is the active conformation where the spike protein stands like this. And in an inactive conformation, the spike protein lays like this. In the active conformation, spike protein binds successfully to the S2 receptor and enters into the cell. But in inactive conformation, it doesn't bind to the S2 receptor. It just remains silent so that it escapes from the immune system. This could be one of the hypotheses that it escapes, the coronavirus escapes from the immune surveillance. So once the body is totally, the immune system fights, when the immune system was taken over by the viruses, we need additional help to fight with the viruses. That's where the therapeutic and vaccine development system comes into play an important role. Now I'm going to discuss about multiple therapeutic options and prevention options here. So one is most important thing, lots of people are talking about the vaccine nowadays. What does vaccine do is it doesn't cure the disease, it just prevents it. Before the infection enters into the body, it prevents it. And in a minute, I'm going to discuss how it does and give some examples. And in the treatment, this is what helps to cure the disease. Antiviral therapies, antiviral agents, or antiviral drugs plays a very important role in fighting the virus to help the immune system to clear out the virus from the body. So how does the vaccine work? Vaccine is nothing but is something like a virus. It's a virus. It could be a virus or a virus-like particle or a substance from the virus or a particle that has a virus substance. When it is administered into the body, it will create an immune response where antigen presenting cells, basically it activates the adaptive immune system. The virus, the vaccines basically activates the adaptive immune system. Antigen presenting cells activated, and then they finds the antigen molecules from that particular vaccine, and then activates the B and T cells from the adaptive immunity, which generates the neutralizing antibodies and various different types of other antibodies, and generates the memory cells, which remembers the characteristic features of the vaccine, which includes the virus, and fights with and this stays there for months to years and years and years, and fights the infection when it enters into the body successfully, not to get the person infected. There are various different approaches developed to create the vaccine, which I'm going to discuss with examples in a minute. But to develop a vaccine, one most important thing is antigen representing molecules are required from that particular virus. In case of coronavirus, spike protein, structural proteins play a very important role to create the vaccines, which is spike protein, envelope protein, matrix protein, and nucleocapsid protein. As of now, approximately 149 potential vaccine candidates are under development all over the world. Collaboratively or independently, various labs are working. Among 149, 25 of them already in the human clinical trials. The very first approach is using the viruses itself as a vaccine. There are two methods here. One is live attenuated viruses, using the same virus, but it is attenuated or weakened. For example, what it does is, in generally, when virus passes through the generations, it mutates a lot. It could be a good mutations or bad mutations in the virus. For the virus, when virus has good mutations, it becomes more and more stronger. When it has a bad mutations, it becomes weaker. So the researchers take this virus, infect into the animals, and passes them multiple generations until they find the inactive weak virus, and which successfully activates the immune response. Here are some examples. Smallpox, MMR, chickenpox, influenza viruses were successfully developed in this method and being used currently. And there are some companies like Indian Immunologicals, Serum Institute of India, Codagenics are independently or collaboratively working on using this method to develop a vaccine for COVID-19 or coronavirus. The second method is inactivated virus. Using inactivated, the live virus can be inactivated using either chemicals such as formaldehyde or heating at certain temperature, or evolving them in chicken eggs. Multiple different methods. Inactivated virus can also create a successful immune response. Here are some examples: polio, hepatitis A, rabies. All these viruses are created using inactivated virus method and successfully being used in, currently. There's two companies, Sinovac and Dynavax, are trying to develop a vaccine for COVID-19 using this method. But all these companies that develop the vaccines that are being developed are still at the preclinical stage. So hopefully they may go towards the clinical stage, and then we will get a successful vaccine in future. The second method is using the viral vectors as a vaccines. Second approach, viral vectors as a vaccines. Here also there are two different approaches. The first approach is replicating viral vectors. This is almost similar to the inactivated virus. So use the inactivated virus, take out all the genomic information of a particular virus and incorporate the genomic information of the virus that you need. For example, in coronavirus, spike protein gene is incorporated into inactivated virus and to generate the vaccine. This is one of the methods. Second method is non-replicating viral vectors. So adenoviruses are best examples. In here, all the genomic information will be removed, and this cannot be replicated. The virus, this method, they can replicate. The virus can go inside the body. This vaccine go inside the body and replicate more, make more and more vaccine to create a stronger immune system. But these non-replicating viral vectors cannot replicate themselves. Whatever we give in the body, that will create the stronger immune system and successfully creates the memory cells in antibodies in the body and to create the immune response. The CanSino Biologics is the first company who started working on the COVID-19 coronavirus vaccine. Before I go what they did, before I go and explain about what they did, I would like to give a brief overview about the vaccine or therapeutic development, the process, the stages in the vaccine and therapeutic development. There are basically two stages. One is preclinical, and the second one is clinical stage. In the preclinical stage, the vaccine or drug will be developed in the lab and tested in the human cells, then animals, then probably a couple of healthy individuals. Once this is cleared the preclinical stage, then it goes into the phase 1 clinical trials, where it will be tested on healthy individuals, approximately around 100, for a safety issue. Then go to the phase 2 clinical trials, where safety and efficacy will be tested on multiple hundreds of people. Then goes to phase 3, where thousands of people will be tested, including the potency, the safety, efficiency. Once this is done, the regulatory process will be taking place, where they decide whether it can go to the phase 4 or not. Once it is phase 4, this can be released into the market as a restrictive drug, as a investigational drug or investigational vaccine to monitor the long-term side effects. All this process takes approximately 12 to 15 years to develop any potential drug candidate or a vaccine candidate. So CanSino Biologics started working on the COVID-19. They used genetically engineered adenovirus 5, a vector. They used spike protein genes engineered with this virus vector with the spike protein gene, and successfully completed their preclinical studies within 3 months. Usually preclinical studies takes around 5 years, and they completed within 3 months. That's a rapid speed. That's like a rocket speed. It's amazing. Then they moved into phase 1 clinical trials in March. The results came out. The results are kind of mixed, because 80% of the participants, the volunteers, reported at least one major side effect, including headache, muscle pain, fever, fatigue, etc., and etc. Right now, they are kind of working hard on making this much more effective, more safe. Currently, it is under phase 2 clinical trials. The results are not yet known, but hopefully soon they will come into clinical trials. They'll successfully complete the phase 2 and go into the phase 3 clinical trials. Another company, this Immunity Bio and Non-Quest, together, working together to develop another vaccine, potential vaccine candidate. They're also using adenovirus vector based vaccine, but this is also an 85 vector, and they're using 2 genes from the coronavirus, which is a nucleocapsid and spike protein gene. They're right now in the preclinical stage. The results may come out anytime. They may go into the phase 1 clinical trials within like a couple of weeks. So let's hope for the best for this vaccine candidate as well. Another big company, which is AstraZeneca, that's also using another genetically engineered adenoviral vector. They're using a vector from a chimpanzee, the adenovirus from chimpanzee, but the other 2 companies are using the human adenovirus. So basically what they're doing is they're working together with Oxford University and a government agency from the United States, which is called BARDA. They have successfully completed their preclinical stage data. Now they're simultaneously running phase 1 and phase 2 studies. So that results may come out at any time. Initially, they plan to move towards phase 3 clinical trials in September, but right now they discussed with regulatory authorities to move even faster, to create a better, to move forward very fast towards the phase 3 clinical trials. Simultaneously, AstraZeneca also working on production side. They are planning to massively produce, once the vaccine is ready, approximately 2 billion doses globally next year. Hopefully, this vaccine candidate may get a success soon. Johnson & Johnson, another big company, they're also using adenovirus, genetically engineered adenovirus, which is 8026. They're also using the spike protein gene to generate the vaccine. They're working together with their sister company, Janssen, and the US government agency, BARDA. Currently, they're in the preclinical stage, but they're probably, again, they planned their phase 1 in September. They're moving into July right now. The results may come up at any time, and then hopefully they're going to move faster towards the phase 2 and then other clinical stages to get a success. And these are also, I mean, this company also working simultaneously and producing massively to supply all the population throughout the world, approximately 1 billion doses globally over the next year. However, there is a risk factor in developing these vaccines from these adenovirus, genetically engineered adenovirus, because so far we don't have any success using this adenovirus vectors to create a vaccine. That's one caveat, but let's hope for the best. One of them may, or multiple of them may get a success to create a successful vaccine to fight successfully with the COVID-19 infections. Second approach is using the nucleic acid-based vaccines, either DNA direct. So in the COVID-19 case, the spike protein DNA will be injected directly into the cells, which translate into mRNA that will generate transcribes into mRNA, which will be translated to spike protein, potential antigen, which will be recognized by antigen presenting cells to create an immune response. The RNA vaccines also possible. The RNA cannot be directly injected, which need to be formulated either in lipid nanoparticles or in polymers and then injected into the cells. RNA will be translated into spike proteins and to create an immune response. In Vivo Pharmaceuticals is one of the front runner company who is working on DNA-based vaccine towards the COVID-19. They have successfully completed the preclinical, which is their vaccine name is INO 4800. They successfully completed their preclinical studies. Currently, phase 1 clinical trials is ongoing. The results may come out at any time. In July, they're planning for phase 2, moving forward to create move forward with other phases as well. And one more important thing in here is, this is the only company so far generated or developed a vaccine for MERS coronavirus that is currently in the phase 2 clinical trials. And they also have a proprietary device that administers the virus through skin straight using an electroporation method. So fast, easy, and simply. Another company, Moderna, which is working on an mRNA-based vaccine, mRNA 1273. So what they're doing is they're taking an mRNA that codes for spike protein, encapsulated in the lipid nanoparticles and injected in the cell, which generate the immune response. They also successfully completed a preclinical stage and almost the phase 1 as well. Currently, they're doing the phase 2. They're probably in a month or two, they're going to go for a phase 3 clinical trials. These are this is moving a bit faster than the other vaccine candidate development. But initial results are a little bit, they found very good results in the phase 1, good safety studies. But they're also working hard on making this successful. And in this approach also, there is a risk, which is so far we don't have any vaccine developed using either mRNA or DNA based for any other virus types. But let's hope for the best. Hopefully, mRNA or a DNA vaccine will be successfully created by end of this year to save the people from this COVID-19 infections. Another approach is protein-based vaccines. So using the protein itself, in coronavirus case, spike protein or a subunit from the spike protein, like say S1 or S2 subunit, or a domain of the particular spike protein, like receptor binding domain, will be formulated and injected into the cells, which will create the immune response. Novavax is the company that is working on the creating protein-based vaccine. They are using the spike protein, encapsulating into lipid nanoparticles, which is a proprietary for them, which generated from plant lipids, which is called matrix. And they have successfully completed the preclinical studies. Currently, phase 1 studies are ongoing, which may complete at any time now. And they're planning to move towards the phase 2 clinical trials in July. Hopefully, this may give some good results and then get a vaccine for it. So now I'm going to talk about the therapeutic agents, developing the therapeutic agents, which once the virus enters into the body. There are two approaches for this one as well. One is repurposing of existing therapeutics. The second one is developing the new therapeutics based on the genetic information from of the virus from the scratch, from nothing, which takes a longer time. But the repurposing may be faster because we're going to use existing broad spectrum and existing broad spectrum of antiviral agents can be screened, or the other drug molecules which can cure other diseases like blood pressure or fever or heart diseases can be screened against the coronavirus. And one of the potential candidates, we may find some potential candidates which will be much more better and faster because they're already approved, completed the clinical trials and approved by the FDA. Here I have listed several candidates, antiviral agents that came out of the repurposing stream. Except remdesivir, all of them are approved by FDA against various different viral infections. These two, lopinavir and ritonavir, are protease inhibitors. They are approved for HIV infections. They also showed some promising effect on coronavirus proteases as well. And these two, imifenavir and neratinib, are approved for influenza and hepatitis C viruses. This molecule shows some activity on the spike protein of the coronavirus, and this one showed an activity on the nucleocapsid protein. And these three molecules are inhibiting the replicase enzyme in coronavirus. But the favipiravir and galidesivir, these two molecules are approved by FDA to treat Ebola virus infections. Remdesivir is in the development to treat the Ebola virus infections. When it was repurposed towards the COVID-19 infections, it showed some promising results, which I'm going to explain you in a bit. So the favipiravir basically blocks the replicase, and here is the structure of the favipiravir. Once it enters into the body, it will be modified by the body in cellular enzymes into an active form. This is successfully completed all the three clinical phases, including a preclinical study. Right now, it's a phase 4 clinical study. Last week, Glenmark announced that they are bringing it into the market in the brand name of Fabiflu. Again, this molecule is approved by Drug Controller General of India as a restricted emergency use medicine. This is not completely approved. It's as a restricted and emergency use on mild patients with mild to moderate symptoms, so that they cannot get to the severe stage of the infections of the disease. Remdesivir, this also a potential candidate that blocks the replicase activity. Initially, it is developed by Gilead Life Sciences. Here is the structure of the prodrug molecule as it is made. Once it is enters into the body, this is the active form of the drug, which mimics a nucleotide adenosine of the DNA. And there are some structural differences over there, but this molecule successfully completed preclinical to phase 3 clinical trials as well. In fact, this molecule is approved by FDA in the United States and Drug Controller General of India in India for an emergency use to treat severe symptoms with COVID patients. Gilead is commercializing it in the US. Cipla and Hetero announced last week that they're also commercializing, bringing into the market with this brand names Cipremi and Covinafir in the Indian market. And this is a crystal structure of the replicase enzyme with the remdesivir molecule in there. But still, the thing is, we don't know how this inhibits the virus replication. The mechanism is not clearly known, but the structure, the crystal structure was available. So that means it's binding to the target. That's a good sign. So it's acting on the target. That's a good sign. So hopefully soon it will complete its phase 4 clinical trials. So we will have a good potential candidate to treat the infected patients. This is another novel molecule, EIDD2801. This was also targeted to develop against the Ebola virus infections by Emory University. Now currently it is working together with Ridgeback Bio and Merck pharmaceutical companies, which also blocks the replicase activity. And one of the advantages of this particular novel molecule is it can be taken as oral pill. As the remdesivir, you have to administer either IV or an injection. And this molecule is a crystal, the structure of the molecule is here, and which mimics the uridine and RNA base in the human body, which successfully completed a preclinical trials. Currently in phase 1, the results may come out at any time, move towards the phase 2 and 3. So hopefully this could be a successful molecule to treat the COVID-19 infections. There are some other methods also being developed, such as hyperimmune therapies, using the neutralizing antibodies developed by the body, the immune system. This is what my lab is working on right now. I cannot give the data or any other details because of the confidentiality and conflict of interest issues, but I'm going to give you a glimpse of what we are doing in the lab. So this is the crystal structure of the spike protein, and this is the receptor binding domain. The neutralizing antibodies successfully bind to the receptor binding domain and blocks the interaction of this receptor binding domain with ACE2 inhibitor. Here is one of the antibody, CR3022, that binds to receptor binding domain, is a crystal structure. But the problem with this antibody is it is binding to the receptor binding domain, but it's not in the place where it's supposed to bind, where to block the ACE2 interactions. What we do is we take these type of antibodies and we modify them using some computational algorithms to make them much more potent, and then test them computationally and synthesize them in the lab and test them on the viruses. This is what we do in our lab. So far, I have discussed about the evolution of the coronavirus and towards the COVID-19 pandemic. Then I have discussed about the immune surveillance system, how it fights with the virus infections. Then also I have discussed about the various different methods with examples to make the vaccines, current progress on them, and the therapeutic agents as well. However, so far we do not have a vaccine candidate or potential vaccine for any of these three, SARS-1, SARS-2, or MERS coronaviruses. Because MERS coronavirus is there from past 80 years, and SARS virus is there from past 18 years. Still, we do not have a vaccine. So, how to survive? What can we do to survive this from these infections? As of now, what we can do is we have to live with virus, live like we are living with the virus, and avoid to expose it to it. So these are the cartoons of I'm a chemist. So a chemist's perception of explaining the importance of social distancing, using the personal protection equipment like masks or gloves, and maintaining the personal hygiene, like using the sanitizers or cleaning the hands frequently with the soap water. So don't go out, stay home. This is a homo chemistry. Students may understand better about these cartoons. Stay home unless until it's really necessary to go out, which protects you from virus infection. And these three are eating well, sleep well, be active, which basically strengthens the adaptive immune system. Eat well means nutritional food, and do some exercise activity, which helps your adaptive immune system to strengthen. In this cartoon, basically, this is an eclipsed form of ethane, where one of the carbon is wearing the mask, the other carbon is not. So this carbon is yelling at the other carbon, where is your mask? And after a few minutes, the carbon takes the mask, and in here also this hydrogen yelling at is, why are you so close? Stay away. So maintain the social distancing. Then within a few minutes, the carbon rotates in a 180 degrees, which forms a staggered form, and the hydrogens are distanced enough. So this is explaining a chemistry way of social distancing. So in this journey, there are many people helped me, including my students, my colleagues, my mentors, my faculty members from all these prestigious institutions. My journey, chemistry journey, started from OU campus, Department of Chemistry, where I started learning chemistry. Then I worked briefly at the Indian Institute of Chemical Technology, CSR, IICT, where I started my first reaction. Then I moved to University of South Carolina for my PhD. I was trained as an organic chemist here, then moved to Wayne State University for my postdoctoral studies, where I learned molecular biology while I'm implementing my organic chemistry skills. Then my first job at MIT, where I started learning some cellular biology techniques while I'm implementing synthesis and molecular biology techniques. Presently, I work at Expansion Technologies, where I'm working with human organs right now, where we are developing early stage diagnostic methods for various different cancers, including prostate, breast, pancreas, liver, kidney, etc. So I would like to wholeheartedly thank all my friends, colleagues, students, mentors who helped me in this my journey. Thank you so much for your attention and patience. I apologize. I think I went about 10 minutes over, but I appreciate your patience. I'm happy to take any questions if you have. Thank you. Thank you so much.
Hello. Okay. Thank you, Dr. Mahender, for your insightful presentation. So now we will open this webinar for a question and answer session, or any comments or anything if you are interested, participants can raise their hand option, or they can chat. They can ask question in the chat box. So anyone can unmute themselves and ask question. Hello.
Yeah. Open to the questions and answers.
Hello.
Yes.
Yeah, yes, sir. You can
Good
go
morning,
on.
sir. Good morning.
Yes. Good morning. Good morning.
This is Dr. Madhavara. So actually that COVID-19 is the non-living protein part. Can you destroy that protein part by denaturing that?
Denaturing on virus?
Yeah,
Yes,
it is a
any
non-living protein.
can
Protein can be destroyed.
Yes, yes. See, a protein can be denatured. That's what you can. If you see one of the method that is um, inactive virus. So basically, they use heat inactivation to destroy the viral part. The virus. It could be proteins or it could be any functioning genes. But the problem with the coronavirus is the spike protein is very important. It has to be active to generate the immune system. Right? So it's kind of hard to do anything with the spike protein unless we have to use it like a bait to activate the immune system. But again, I mean, it's possible.
So,
Sir, that was a nice presentation.
Thank
I
you.
have a general question. We have
Sure.
heard the possibility of reoccurring infection after the recovery. I would like to know about that. And is there any possibility that our immune system might respond positively in the second occurrence?
Yes. So that's a very good question. There's a lot of discussions on the, a lot of research is going on into it. So it's called immunity passport. So if he is protected, like a person who already successfully recovered from the COVID-19 infection, if he can be protected from the second time infection, that's an immunity passport, which is a very big question mark, because what recent studies are showing is that the neutralizing antibodies developed by the adaptive immune system are only surviving a couple of months, not more than beyond that, which indicates that he's kind of prone to the infection again. So there is a possibility in there. It all depends on how strong the immune system is developed, the neutralizing antibodies or memory cells in the body. But there is a possibility.
Yeah, then the government should start testing them again, right? I mean, they are
Absolutely.
releasing. Yeah.
Absolutely. I think, I mean, once the incubation period is over, probably they're being released. But once in a while, they have to go for additional testing to, I mean, confirm that, I mean, well, if they get the symptoms again, obviously they have to go for a testing, right?
Yeah.
So
Thank you, sir.
Hello, sir. There is a question in the chat box. So very nice and informative talk, sir. Can you please comment on the plasma therapy, considering rejection or autoimmune reaction into picture?
yeah, it's a very great question, actually. So the plasma therapy it's an experimental therapy because each individual's body generates various different types of antibodies, neutralizing antibodies, right? So it varies. It may work for certain individuals. It may not work for certain individuals, and it may be generate some autoimmunity on certain individuals. And so that's why it's an experimental medicine. They're trying it from hundreds of years. This plasma therapy is there from hundreds of years, trying for various different diseases and cancers. It's an experimental. So it's like, that's why the bone marrow or plasma, the blood plasma will be taken and injected to see if he recovers. That's he recovers. And then we have some data that, X amount of people are recovered. Some of them are not recovered. I hope I answered that question.
So there is a question.
Go ahead.
Yeah, recently there were talks about like O positive blood group are very resistant to coronavirus. Is that true? Or how does that affect the coronavirus
Yes.
actually?
Yes. So there is a couple of studies published recently. Again, this virus is very new. Everything we are knowing day by day. Every day something new news comes out of it. So there is a very nice, interesting study was published in very high impact journal saying that O positive, O group blood is kind of producing is more protective compared to the A group blood. So AB, these three blood groups were tested. So AB compared to AB, O is much more protective towards the COVID-19 infections compared to the AB. It could be, O group people are generating probably such more potent antibodies or memory cells. That could be possible, but how, why and how is not quite clearly known.
So there's another question.
Go ahead.
So recently I've seen some government official was saying like asymptomatic cases they are from asymptomatic cases airborne transmission is not possible transmission. If we find asymptomatic cases soon and if you track them down, then we can restrict the virus. Is that true? Like,
so if I understand correct, so the transmission from asymptomatic patients is not possible. Am I correct? That's what is the question?
yeah. That's what he said,
Yeah,
and I have doubts on it.
Yeah. So the thing is, there's no clear data available that saying, it's not the asymptomatic people cannot transmit the virus. I'm sure, I think, most of the people, most of the hospitals, doctors, scientists saying that, yes, still they are transmissible. The asymptomatic or non-symptomatic patients also. It's quite, some of the data suggesting that, they are transmissible because the virus is inside of them. It can come out somehow. The symptoms they're not showing the symptoms. Maybe their immune system fighting pretty strongly, very well. That could be a reason, but definitely, I mean, they're certainly, infectable to other people.
So there is a question from Srikant. Most of the viral infections are autoimmune disorders. Why corona is so problematic?
It's not quite autoimmune disorder. It's a fight between the virus and immune system. The autoimmunity, basically what happens is the immunity itself harms the body. It's not like that. So coronavirus probably, diverting the immune system to against the body possibly. It's not quite autoimmunity. Okay, so it generates like it generates some wrong signals, sends wrong signals to the immune system, which creates lots of cytokines, inflammatory cytokines, which cause the severe disease in the human body. But I don't think there is a correlation between autoimmune versus the viral infections.
There is a question from Navya, who is doing a PhD in animal biology and biotechnology in University of Hyderabad. Is there any effort towards purifying the antibodies from recovered patients and production of the same after a promising efficacy?
Yes, there's a lot of efforts are going. One of my lab is working as well on that. So many groups all over the world identifying the neutralizing antibodies from the convalescent patients. Convalescent means recovering patients, and testing them against the pseudoviruses and real coronavirus in the lab for the efficacy. The problem is each individual produces different types of antibody, right? There's so many of them. And the therapies, the way they're creating therapeutics is like an antibody cocktail. Multiple neutralizing antibodies together may successfully neutralize the virus. So that's another approach. And or individual antibody may successfully neutralize the virus. That's another approach. Yes, people are certainly purifying them in the antibody, like even not only purifying, but modifying them as well. Like I said, using computational methods and then resynthesizing them.
So I have a question. There are cases coming up from Andhra Pradesh. So there is no symptoms. There are no symptoms, but there is immediate fatality rate. So asymptomatic but immediate fatality. So that is what
Yes.
coming up in Andhra Pradesh.
Yeah, some of the cases are kind of strange. This still lots of studies, a lot of information we need to know about this virus. How this is happening is not quite clearly known. Lots of people are trying to understand that what is happening inside the body. Even some healthy individuals also being killed by this virus. So some of the malfunctions happening in the body, like fighting the virus. And what is it? It's not quite clearly known.
There is one more question that is coming up in the chat box. Is there any efforts towards, since the clinical trials and getting them into market is a time-taking process, considering the current scenario, is drug repurposing the only quick solution available, sir?
Yeah, that's why lots of efforts are putting towards, and then you can probably see that couple of potential candidates are already coming into the market as an emergency use. If patient is critical situation, we can take his permission or his family permission to administer that particular drug so that it may save them. Again, the stage 4 clinical trial is the one that takes longer time to see the long-term side effects. That's obviously is going to take months to years. But yeah, repurposing is one of the fastest way to develop or to create a potential therapeutics for any type of infection.
So there is a private question. It's something on post recovery. So coronavirus is also impacting the other body parts. So there are research that are coming up. It's not only the respiratory system, but it is also impacting other body parts after recovery.
Yeah. The thing is, this ACE2 receptor, the ACE2 receptor is most commonly found in lungs, kidneys, and heart, and some other parts as well. So there is a possibility that the virus may travel through blood and then go to these particular parts and then infect those cells in those particular parts and start dysfunctioning those organs. That is a possibility, right?
So anyone else who wants to ask question, they can unmute themselves and ask. So Hello. So I think there are no questions, right?
So one more question.
Go ahead, Mr. Akin.
So what is this virus? Does virus mutate itself?
Yeah, yeah. I
Been
mean,
seeing like
see, the mutations happens, in the DNA or RNA, right? So when enzyme start making more and more RNA, basically it's like a chain reaction. It adds bases to one after one, one after one. There is a possibility that enzyme can make a mistake. Instead of adding adenosine base, it can add a guanine base or a cytosine or another base, right? That's how mutations happen. That one single letter changes entire downstream process, changes the amino acid sequence and structural properties of the protein, lots of things. The one single base in the DNA or RNA isn't sufficient to create a lethal or inactive mutation of a virus, or even in the human body. That's what I mean. Like cancers, mutations are the cause for most of the cancers. In mutations in the genes, the human genes.
Yeah, there is a question from Sumita Garu. What might be the reason for high mutation rate in COVID-19?
It's a virus. It's a virus. Any virus you take always mutates. It's a survival, what you call, factor for viruses in general. They constantly mutate
Sir, there
to
is
escape.
yeah, there
Go
is a
ahead.
question, sir.
Yeah, go
Actually,
ahead.
I have worked with a project of papaya ring spot virus, which is a plant virus, and we have actually we worked in that we found that it changes from one region to region within the same state, just some square kilometers after the whole that RNA construction is changed. Like some mutations might happen. This is because of the climate
Yes.
change, climatic changes. So, what is
It
the
could be.
how the COVID-19 is changing? Like, is it changing from region to region, or it's all same? Like, what is the mutational rate?
Good question. Very good question. Yes. See, mutation can happen for several reasons in general. Like, a chemical can cause mutation, a radiation can cause mutation, or UV light can cause mutation, or certain molecules like some poisonous molecules can cause mutations in the body, right? Same thing. It could cause region to region, person to person. You can see different mutations in the same family, two persons in the same family. There's a possibility. That's why you're calling it even region to region. Now, even for the COVID-19, for example, it varies between different continents. Even. The patients that have mutations in China doesn't have the US. The patients in the US or in Europe, it's possible. And genomic databases keep increasing with the new information about the new viruses, new virus mutations every day.
So there is question. There are recent reports on dexamethasone
Dexamethasone.
coming up to repurpose, but it has implication like including glaucoma on long-term administration. How promising is considering such drugs?
See, again, this is happened in the UK. Like it's an experimental medicine. It's just when doctors see they try to give a medicine that can pacify some of the symptoms. That's how they started doing. Like I know a few months ago, hydroxychloroquine was a hot molecule, hot drug to cure COVID patients. But recent studies says that it's not really impacting anything to the COVID patients. In fact, it's kind of becoming a lethal to the COVID patients. Hydroxychloroquine. But this is a steroid. The dexamethasone is a steroid, which reduces the inflammation. Inflammation is what is most important to cause for the tissue damage, right? So probably what's happening in there, nobody knows. Again, this is administration of the dexamethasone is helping patients to recover from ventilators or severe disease. That's what is the data from, I think about 2,000-3,000 patients were tested in UK. And most of them, 80% or 70% of them kind of recovered. Probably some other people are, trying to understand this, like what people are trying to understand on hydroxychloroquine from past 3 months and then realized what's happening with it. Maybe in a couple of months, we will know what dexamethasone is doing as well.
So there is a question. I think
Yeah.
East Indian COVID-19 strain is different. Are they turning dangerous?
The answer is maybe or possibly. You cannot divide a strain between the countries. It's a virus. It always replicates, right? It may become more potent. Maybe some of the patients in India are kind of very good hosts and creates a stronger viruses, very infectious viruses. That is possible, but we cannot generalize that as in, like, in India, COVID-19 strain is much stronger than the other. We cannot generalize because you see, people are dying all over the world now. I think US is number 1 and India is number 4. Russia, Brazil are number 2 and 3, I guess. So we cannot generalize that in a particular country, a particular continent, particular reason at this moment. Like, even for example, like if it is a epidemic like MERS coronavirus or SARS-1, probably we could have had like, some kind of idea. But this is a global pandemic, all over the world, everywhere it is spread out.
Oh, so another question.
Go ahead.
At first, we have thought that India has more temperature in summer, so it can control the virus. It was I know it is not the case now, but I would like to know what is the temperature effect on this virus? Is there any?
So if you have seen my slide where I discussed about the surface stability of the viruses and various surfaces at various temperatures, right? So 37 degrees centigrade, up to 2 days it can survive. And in India, how much it can go? To max 40 degrees centigrade, right? In summertime, 40, 42, 45 maybe. It can survive probably a couple of days, I think 1 or 2 days maybe. But at 70 degrees centigrade, the virus is cleared out within 5 minutes. So I don't think the Indian temperature can go up to 70 degrees centigrade, right? I don't think anybody will survive at that temperature. So I wouldn't say the summer in India helps to kill the virus. Maybe a minor, maybe a little bit small effect could be there because of the heat. Yes, there could be a small effect, obviously, but it's not a drastic impact. That's not going to show a drastic impact. And we should not think like, hey, okay, go ahead.
Sir, I have a question. This is related to herd immunity. We are finding,
Yes.
for I think last 2 days there was a warning from WHO on Swedish policy on herd immunity, where Swedish epidemiologist Anders Stagnell is supporting the herd immunity policy of their country, but WHO is saying spike in the next wave in the Sweden. So what is your take on herd immunity?
Yeah, what I think I recently heard about this herd immunity, and it's a theory. Herd immunity is a theory. It's basically, I mean, in the Swedish, it may be true, but the data, I don't know how to interpret the data. It's based on the probability. For example, let's say in your house, there are 10 people living. Out of the 10, 6 people are kind of more protective towards the COVID infection, 4 are less protective, right? If these 6 people go outside, come back home, the probability of the other people getting infection is kind of very low, number 1, right? If anybody from these 4 more infectious people go out and come back, the other 6 people getting infection is kind of, a probability is kind of low, but rest of the 3 people getting infection is probability high. So this is basically a probability theory. That's what I understood from herd immunity. But how practical it is, I'm not sure, only because lots of people like New Zealand, Sweden, those countries effectively control the virus infection, right? Probably based on that, they're coming up with these theories. But to me, it's a theoretical hypothesis.
Yeah, there is a question again, but HCQ is effective against parasites, while this is whole new class of pathogen. Maybe we cannot rely on such drugs in the first place. Please correct me if I am not right with my assumptions.
Yeah, you're correct. Actually, it's a malarian drug. It treats malaria, but it also has severe side effects. That's the big problem with the hydroxychloroquine. And that's why first thing, lots of repurposing studies have done. Later, selective studies also has been done, how it is impacting the coronavirus. No information is available. People think that it is probably attacking on multiple different targets, maybe spike protein, nucleus protein, replicase, or anything. Randomly, probably it is acting, but I think rather than curing the disease, its side effects are more impacting the patients compared to the COVID infection. So that's why it's kind of now lots of companies are dropping off clinical trials using the hydroxychloroquine on patients. In fact, I think last couple of weeks ago in UK, they tested on 5,000 people. Lethality rate is very high on those 5,000 patients. So that's why they immediately stopped using this HCQ.
Yeah, I think it's already 11:50. I think we'll close this session, and we are very grateful to Dr. Mahender Dewal. I think it's around 2 o'clock.
My
there.
pleasure.
Yeah, it's 2 o'clock there, right? At night. So
2:30 almost.
2:30. Yeah. So we are disturbing your sleep. So
No
we
worries.
are.
No worries.
Yeah.
It's my pleasure.
Yeah. We are very thankful to you, sir, for giving this insightful talk, and we are looking forward. And there are so many questions, very interesting questions coming up with.
Ask me offline. I will answer all. Send me to my email.
Definitely.
I'll send you all the responses. No worries.
Yeah, you please,
Thanks
anyone
for
can,
hosting me
if
today.
you have any questions, they can email us or ask in Facebook also. They can send message. So we are open.
Yes, on the
And
Facebook as
with
well.
this,
Yes.
yeah. With this note, we thank everyone, every participants. We actually, in fact, got registrations from different universities across the country, and across maybe even some in US also. Some students from maybe friends of Dr. Mahender also registered for this. And this video will be uploaded. It's already there on Facebook. In our page, you can also share it or inform your friends or family members or anyone who actually interested to watch. So it's open. So with this note, I thank everyone. Special thanks to Dr. Mahender. Thank you. Thank you, Anand.
Yeah. Thank you so much, Kiran, and your team. Thanks for hosting me today. And also, if you have any questions in future as well, send me through either Facebook or email and contact Kiran. He can contact me as well. So feel free to ask me in future questions as well. Thank you. Thank you so much. I appreciate your time and patience.
Thank you, sir.